know, and I'm sure you understand, like what would our, like for example, pre-existing heterozygosity, like I understand you don't agree with it, but what do we mean by that when we say that?
That there are genes that exist on, different chromosomes that we replicate via, that we can't replicate on our own, that we require the use of another human being to replicate.
Okay, yeah, so like millions of differences, say in the DNA sequences of chromosome pairs. We're saying that that was built into organisms from the beginning by the creator. Okay, so when we look at the origin of species, we're saying that long periods of time is not required for the new species, because if we start with these heterozygous organisms, the visible distinctiveness and change in offspring, it's going to be fast. So if you look at things like recombination, gene conversion, that would provide many new varieties of, say, combinations of traits quickly, because we're saying these differences are already built in. So if we go back to functional versus non-functional, non-protein coding regions, we predict that, they're not going to be idle. They're going to have function, because if we're saying that these tens of millions of nuclear DNA differences were built in, if they're design variation, we'd expect them to have action. Okay, so obviously ENCODE revealed that 80% is transcripted, and now we're seeing more function. Like for example, pseudogenes, which we thought were false genes, actually show function, or something like protein moonlighting. We're seeing these proteins that have multiple function. That's consistent with us, because that's a prediction, a testable, falsifiable prediction based on pre-existing heterozygosity. But you're saying, obviously, these are not true functions, or it's spurious transcription. To me, that's a rescue device. How do you feel about that?
So it comes down to how you view something as functional. I mean, if you're considering the fact that one gene is transcribed in the course of, 8,000 runs as being functional, sure, that isn't really a valuable definition of functional, because it means that at best that, okay, there's a gene transcribed, but it's far from being transcribed regularly. There's some genes that are transcribed, 30, 40 times within an hour, let alone 30, 40 times within however long the duration of the experiment is.
So are you familiar with Mendel's experiment on the pea plants?
Yeah. The one thing Mendel proved is what he got wrong.
Well, he proved, because he was looking at the principles of inheritance, right? So he found concerning, and correct me if I'm wrong, but he found concerning traits that they actually remained distinct, as in they didn't blend, but they appeared and disappeared in family trees, right? They came in dominant and recessive types. So what I'm saying is,
you are familiar with the history of Mendel, though, right?
Well, I know him and Darwin. He didn't know genetics, and neither did Darwin, but he's seen this based on the experiment he did.
Are you aware that Darwin also did experiments in genetics?
No, but he didn't know of he didn't study genetics. Everything, obviously, genetics came about. The field of genetics came about after both Mendel and Darwin.
Sure. I mean, Mendel is considered one of its founders. However, even with Mendel, part of it, the fact is that when other people studied Mendel's work, they noticed that it was almost instantly incorrect, because there's information that was left out.
Okay, I just want to clear something up. You agree with me that Mendel and Darwin were ignorant of genetics, though, right?
Sure. I mean, genetics was just becoming a thing. It was in its infancy.
Okay, so he did see, though, that these traits, they appeared and disappeared in family trees. They came in dominant recessive types. But now that we look at a genetic level,
sure,
we know why that's to be. Is what I'm saying, that this is consistent, and it is indicative of the pre-existing heterozygosity that we believe in regards to the origin of species. Because, like I said, heterozygotic DNA sites will provide that necessary variation. We look to Mendel, to provide the necessary evidence for that. Obviously, that's all I'm saying with.
Well, there's a lot of things wrong with the experiments he did. The first of which is the fact that, one, he ignored evidence, and one of the evidences he ignored is the fact that a very specific amount that would be predicted by what we consider modern, Mendelian genetics, is that when he breeded certain plants, there were hybrids between the original and the parent, which is the same thing Darwin observed. And also, what was failed to be noticed by Mendel, and couldn't have been known at that time actually, was the fact that he was particularly examining a particular thing in genetics.
Okay, I just want to stop you right there. Are you saying that his pea plant conclusion based on these traits, recessive and dominance, that's not universally accepted? Like if we look at the pea plant conclusion, are you saying that's not universally accepted?
The result
because we're focusing on the pea plants here.
So when he published his results, they were not initially accepted.
I understand that because they didn't know genetics back then.
Well, people had been doing these experiments for a little bit before then, and they understood kind of the rough aspects of it. But people didn't accept it for a few reasons, and one of those reasons was that he concealed data, and the other reason is that when he was dealing specifically with a phenomenon in genetics known as independent assortment.
Okay, but are you saying that what he observed in regard to family trees, with the traits, are you saying that that's not, like, I just wanted to, do you agree with that part?
So the mechanism he observed within pea plants is highly non-characteristic of many organisms that we do see.
Okay. So what, and give me a comment on this, what Mendel proved and is today accepted, I'm saying this is accepted universally, is that a new trait can emerge in just one generation based on his pea plant and pre-existing differences. So we're saying if the created kinds and Adam and Eve had these pre-existing differences and pre-existing heterozygosity, change in new traits can come about quickly. Go ahead.
Yes, it can. However, the crux of that is, once again, when he was performing his experiments, one of the interesting things about it, and there's been molecular studies on this experiment as well, because they've recreated it, is the fact that the genes of interest for Mendel were significantly spaced out, which what that means, unfortunately, is even if an organism inherits, there's a very fair chance that an organism will inherit one gene and still maintain the other, its original genes. In most organisms that we observe, however, that's not the case. Most genes tend to overlap with one another. So if I inherit a mutant form of one gene, I'm more than likely to, I'm more likely to take on or to inherit other genes that are mutant.
Well, it just sounds like to me that, you can't grasp pre-existing differences, because obviously in the evolutionary literature, mutations are the source of all differences, right? Because
yes, it does.
Okay.
So the big difference, though, is that if we're dealing with, like we were talking about before, with coding and non-coding, right? Mm-hmm. Genes are regions by definition, where proteins are coded for and have a function. If there's significant difference, like if there's significant spacing between these regions where genes can be expressed, well, of course, with crossing over, you would expect that some genes get passed on, some genes don't. You're going to have some kind of distribution that determines that.
Yeah, but I'm saying with the pre-existing, because would you agree that human chromosomes differ by, say, millions of single nucleotide polymorphisms, like SNPs, between and within individuals? Would you agree with that?
Yeah, I would think so.
Okay, so if we look at DNA sequences, say in a chromosome pair, for example, we know they're, let's say they're identical, okay? They would be said to be homozygous, right? So, but when they differ, even by one base pair, then they're said to be heterozygous, right? So I'm saying inheritance and not mutations is the main cause of heterozygosity within individuals. And regardless of all the details of Mendel, he did demonstrate recessive and dominant traits. So that's all I'm saying. And I think that that is a basis and a starting point for what we're saying in regards to the origin of species.
So here would be my question then. Would you say that there are certain combinations of genes that like, you know, I can have different forms of a gene, but they can all be considered homozygous?
Okay, so you're saying you're going to have multiple variants of the same gene, but at the same time it's homozygous?
Right. So what I'm saying is that it may be possible for, you know, a particular coding region of DNA to be selected for because there happens to be a certain, you know, a certain pattern of nucleotides that favors the transcription of that particular gene into this phenotype, as opposed to another gene on another chromosome being expressed due to
Okay,
something like codon specificity or.
okay, yeah, I understand. Okay, so what you're saying is all genes have heterozygotic gene sites and homozygotic gene sites. Like there's no actual population that's just purely homozygous or purely heterozygous, right? Is that what you're saying?
Yeah, effectively.
Yeah,
And
I would say that that's pretty basic. But if we go back to our model with pre-existing heterozygosity, say encoded into each created kind and into Adam and Eve, that speciation coming from there, obviously, we're going to see mutations afterwards, and we're going to see degradation and sorting out of information, and it's going to go from more heterozygotic DNA sites towards homozygotic DNA sites. But of course, there's still going to be both, right? All genes have both.
well,
I agree with that.
the only way you would get homozygotic gene sites is that if humans somehow decided to or started to learn how to lay eggs. I mean, because of the fact that human beings, when we reproduce, our DNA combines with the DNA of another person.
Correct.
So the chance of mutation in that a beneficial, some kind of beneficial mutation will occur is increased because of that, as opposed to in a homozygotic situation, if I'm laying an egg and the DNA I have is just replicating and creating copies of itself. So,
okay.
I mean, as long as people keep, reproducing in a heterozygotic fashion,
Sure.
I mean, we've talked earlier that, sure, you're probably going to have a lot of bad mutations, but you're also going to have a few good mutations that are going to get selected for and passed on through the generation.
But we can get a heterozygous gene site if the animal didn't migrate and get isolated. Because what we're saying is the pre-existing differences, they were far more heterozygous sites than homo at the beginning, right? Because would you agree that a small heterozygous population will tend towards homozygosity? Like when this happens, and this kind of goes back to the Mendel pea plant, example and experiment, some recessive traits will be revealed. And it sounds like the creation and evolution models both agree that all species within families share common ancestors. And we also agree that if you don't, let me know.
Well, so I mean, here's the thing, though, right? Is that like with human beings, there are more than one species of human before we wipe the majority of them out. But things like plants, for instance, things like other mammals, there are other species and there are other classification of phyla that species can breed within.
Okay, well, obviously, I don't.
Like if you're restricting it to breeding within only its own species, you may have a point.
Well, would you agree? Okay, would you agree that the parental generation would be more hetero than its offspring generation?
Not necessarily.
Okay, the majority proves the rule. Would you say most of the time?
No.
Okay, but you just agreed that a small heterozygous population will tend towards homozygosity, but now you're saying that it's not the majority of the time? Like, I don't understand.
So generally speaking, right, in the behavior of animals, usually incest isn't usually incestuous relationships, usually incestuous behavior isn't favored among reproductive successiveness. So usually you will have organisms that, wander a great distance, decide to lay eggs, another species comes along, or another, family of fish that are completely unrelated come along, fertilize the egg, and you have an increase in heterozygosity.